Hello everyone, you all guys are listening me on Unacademy online platform. As you know, it is a biology series for all examination, and here I'll discuss with you a third part of cell biology. As you know, it's a very lengthy chapter, so I just have split it into three parts. So here I'll explain with you the remaining part of cell biology. This is my lesson number 5 in the biology series, and before we start the lesson, I would like to introduce myself. My name is Dr. Ravi P Agrahari. I'm a PhD of IIT Delhi and working as a director of Purvanchal at Gorakhpur. My 2 books are published on the McGraw-Hill, that is highly recommended for all of you for science and tech and environment. This is my Telegram channel that you people can see on the screen, where you can find out all the entire PDF. My Unacademy profile you can also see on the screen. Here you can find out all the plus classes are part of many of the free special classes. So I'll request to all of you kindly follow me here, and this is my code. Whenever you are going to join any courses, if you use Ravi UPSC code, so you will get extra 10% discount in your fees. India's best educator for UPSC CSE, they are taking the classes on Unacademy in the live session. So it is a great opportunity to all of you to connect with all the experienced faculties on Unacademy platform. UPSC CSE Note 2.0, we are providing you with 20 comprehensive UPSC CSE GS booklets for the plus and iconic subscriptions. So kindly subscribe this now as soon as possible and enjoy all the contents. So it's a biological science part 3 of the cell biology, and here that remaining part I'll explain with you. In the previous lecture I stopped with the nucleus, and you people have seen inside the nucleus have a presence of DNA, and a part of DNA, one more nucleic acid you people have seen, that is RNA. 3 types of RNA you people have seen. Now the question is, what are the role of DNA and RNA inside our body? So this is all the entire concept I will explain with you through the help of replication, transcription, reverse transcription and translation. Actually, you know guys, what happened in the case of you people when you are going to form. So very simple, try to understand here. Suppose whenever you people are going to form from your parents, so your parents has given you chromosomes. Everybody know your parents have given you chromosomes, and in the previous lecture you people have seen within the chromosome have DNA. So ultimately in the first day when you people form, I'm telling you as a zygote. So your papa and mama has given you 23 and 23 chromosomes, and within the chromosome have DNA. You know, each and every chromosome have one DNA. It means your papa and mama has given you 23 and 23 DNA. So it means your zygote have a 46 DNA. Notice very clear, your zygote have 46 DNA. You know what happened? This 46 DNA divide and redivide inside your mother body when you people were growing. Okay, that is called as the embryonic development stage. This DNA divide and form a multiple number of DNA. Further, this DNA responsible for the formation of RNA. Okay, this is the DNA to DNA formation and DNA to RNA formation. RNA, you people have seen, they have a different types of RNA as a ribosomal RNA, messenger RNA and transfer RNA in the previous lesson. And these all the RNA as a ribosomal, as a messenger and as a transfer RNA. These all are responsible for the formation of protein. These all are responsible for the formation of protein, and slowly and gradually in that way guys, when you people born, you people have a 3 kilogram of protein, suppose it is your weight. So how our entire body develop in 9 months and 6-7 days inside our mother body. You know, inside the mother body, mother is giving you the nutrition for the development of a baby. Okay, and how the baby is growing inside the mother body, those DNA inside the zygote a baby got from our parents. As you know, 23 DNA got from papa and 23 DNA got from your mama. Okay everybody, 23 DNA you got from your mother, and this is a 46 DNA. This DNA divide from many DNA form many RNA form different types of RNA, and these different types of RNA combine together and form a protein as a 3 kilogram of baby. So I can tell you, whenever you people are born as in the form of some protein character. Your eye is black, so black color eye protein. Your hair is curly, so your curly hair protein. So ultimately, as after the 9 months and 6-7 days, those formation of a baby happen. It is the entire protein structure. Okay, and now how this entire protein structure form? So here have certain procedure. Like in this cases have a replications. Like it is DNA to RNA formation is transcription. Okay, this is what transcription this is. RNA to protein, RNA, this is the different types of RNA. RNA to different types of RNA is called as actually post-transcription. That is not mentioned here. This is called as a post-transcription. This is called as a post-transcription. So this is the replication. This is transcription. After transcription, the next process RNA to different types of RNA formation, post-transcription, and these all the various RNA combine together and form a protein. This is called as this part translation. So what actually replication, transcription, post-transcription and translation, everybody clear? Sometime whenever it's required, this messenger RNA further convert and form the RNA. Okay, this messenger RNA, just I'm telling you, this RNA, okay, don't go for messenger RNA. I'm telling you when RNA is going to back and synthesize the DNA. So it's a reverse transcription. As DNA to RNA formation is a transcription, if RNA further form the DNA, the backside further, so that is called as a reverse transcription. So this is called as reverse transcription. So what actually the reverse transcription, against the transcription, those processes happen, that is called reverse transcriptions. And these all the informations you can see on the screen, those are written now. DNA to DNA formation is called replication, or it is also called as a duplication. The formation of messenger RNA from DNA in the presence of RNA polymerase is called as a transcription. So finally DNA to RNA formation is a transcription. Okay, and the formation of DNA from RNA, is called reverse transcription, as I told you guys. And this concept was proved by Temin and Baltimore. And this is the reason, because the scientist Temin and Baltimore was behind it. This process of reverse transcription also called as Teminisms. And one thing more you people know very well, those RNAs form here, there are these three different types of RNAs, and these three different types of RNAs are responsible for the synthesis of protein, as in the previous lesson you people have seen how ribosomal, messenger RNA and transfer RNA are responsible. Okay, and this process of RNA to protein formation is what? This is called translation. So I hope this flow chart those I made here guys, it will make all the picture very, very clear. Now, inside the DNA have a gene actually. Okay, now the question is, those pictures you people have seen, what are the gene actually? Actually, those 46 DNA you people have seen, those 46 DNA you people have seen, try to understand. Those 46 DNA you people have seen, after 9 months or 6-7 days, have a formation of a complete 3 kg of protein. Have a formation of 3 kg of protein as an individual, as a living body. So it's a 3 kg of protein formation happen. Within the DNA have a presence of gene. You remember guys. And the gene are the hereditary character. Genes are hereditary characters. Actually, within the DNA, your papa mama has given you a gene, and genes are the characteristics inside the DNA, and these genes are responsible for the formation of protein. Okay, and those all the genes are present inside your body. The sum of all the gene is called as genome. But again, I will repeat guys. Gene, this concept, the term was given by Johnson. Okay, Johnson described the gene, it is a hereditary characters, and genes are located in the chromosomes. One chromosome have one DNA. The picture is very clear. Again, I repeat, your papa mama has given you chromosomes. Again, I will revise everything. Your papa mama has given you chromosome. 23 chromosome father side, 23 chromosome mother side. Within the chromosome have presence of DNA. So 23 chromosome have a 23 DNA, and 23 chromosome have a 23 DNA. So that is the reason total 46 DNA you got from your parents. So this is the reason here. One chromosome has one DNA. I hope you understood. Okay, so within 46 chromosome you got from your parents. Within the 46 chromosome have a 46 DNA. Here 23 DNA you got from papa, 23 DNA you got from mama, and within the DNA have presence of genes. Okay, within the DNA have a presence of gene. So this is the entire characteristics, and the gene, those you people got inside the DNA. This is the hereditary characters, and this is the gene, those are responsible for the formation of any characters. Any of the characters, like I'm telling you, your eye is a black eye. So you have a black eye character. Why you have a black eye character? Because you have a black eye genes. You have a sickle cell anemia disease. Suppose you have a sickle cell anemia disease. So why you have a sickle cell anemia disease? Because you have a sickle cell anemia protein, those synthesize. Okay, whenever you are born. And the reason why sickle cell anemia protein synthesize, the reason inside the DNA, those you got from your papa and mama, here have a presence of sickle cell anemia gene. So this is the concept that I try to define you in this case. All characters of human body is an expression of certain proteins. I hope you people have seen. Every character inside your body is synthesized in the form of protein. And this is the protein synthesis are going to be happen as in hereditary characters. But proteins are not going, genes are going. But whenever the gene is going to develop inside your new generations, so it will express in the form of proteins. Okay, now see, actually there are different types of genes on the basis of functional activity. As Wenger was a scientist, he described different types of genes on the basis of their own functional activities. Like some of them are called cistron, some of them are called recon, some of them are called muton. What are the cistron guys? Any function unit of DNA is called as a cistron. Okay, within the DNA chain, any of the function unit of DNA, okay, is called as a cistron. Okay, it is also called as a structural unit. Recon means those genes are responsible unit of recombination. Recombination means a kind of crossing over. Crossing over is a characteristics even responsible for the exchange of genetic material. Okay, mutation, any unit of gene that is responsible as in the case of mutations. The artificial synthesis of gene is a very important question for your objective paper. Remember, was developed by Hargobind Khurana. And this gene those Hargobind Khurana has developed, it was an aniline tRNA. It was an aniline tRNA. That is in yeast, guys. Yeast is one of the fungi, I hope you people know. And they have a 77 nucleotide pairs. So artificial synthesis of gene was done by Hargobind Khurana. Hargobind Khurana also remember because of this feature. One more thing, guys, like jumping or motile or floating genes. That is also very important question for your objective paper. If UPSC, state POs, any of the examination, if they ask what is a jumping gene, so definitely jumping gene means what? Within the same chromosomes, these genes are available. But within the chromosomes, they are moving from one place to another place. So jumping genes, they change their own position. Simple, guys. In the chromosome, within the chromosome, whatever they have their own locations. Their locations they are going to change. So they are moving from one chromosome to other. It is a jumping gene. Okay, this gene was discovered by Barbara McClintock. The gene was discovered by Barbara McClintock. And see, for the jumping gene, this McClintock has got the Nobel Prize. And one thing more, guys, this is also called as a transposon. This is also called as a transposon. Why transposon? Because they are going to transfer their own position. Okay, transfer their own position. So this is the information about the jumping gene or motile or floating genes. What is the pleiotropic gene? Now the question is, what is the pleiotropic gene? Pleiotropic genes are those, guys, where a single gene are responsible to control an expression of more than one characters. If a single gene is going to be responsible for the more than one character, then it's called pleiotropic. Pleo means many of these. Also, pleiotropic genes, a single gene is responsible to control an expression of many characters. Again, I repeat, if a single gene is responsible for the control and expression of many characters, then it is called as a pleiotropic gene. Pleo means multiple. Now, one more statement about genetic code. Genetic code, it was last year, I have seen in 2020-21, MPBSC even asked the question from genetic code. Genetic code was given by George Gamow. Again, I repeat, genetic code term was given by George Gamow. Where you will get a genetic code. The one part you remember, genetic codes are only inside the RNA. So genetic codes are only inside the RNA. Okay, everyone. And see, those RNA, I'm telling you as a messenger RNA, transfer RNA, anyone, guys. The genetic codes are actually responsible for the formation of amino acids. Genetic codes are actually responsible for the formation of amino acid. And what is amino acid? Amino acid is the protein unit. This is the protein unit. Okay, in 9 months and 6-7 days, those 3 kilogram of protein a baby form. Okay, ultimately amino acid form. And the reason behind the formation of amino acid was genetic code. Behind it was a genetic code. And this genetic code is available inside the RNA. So a very simple mechanism, guys. When the RNA is going to synthesize the protein, as through translation process, you people have seen, as through translation process. Okay, so what actually RNA synthesize the protein here? Within the RNA have a genetic code, and this genetic code synthesize amino acids. So within the RNA have a genetic code, and this genetic code synthesize the amino acid, and this amino acid means protein formation happen. So now the more clear picture is what? Actually within the RNA have presence of genetic code or codons, and this is responsible for the formation of amino acid due to translation. And many amino acid form, it is a some specific protein. I hope picture is very clear. The genetic code I am going to explain with you, guys. Okay, it is what? It is a total 64 genetic codes are available. That I'll show you inside the genetic dictionary. And this 64 of genetic codes, they are in the triplets. Triplet means the group of 3 bases. The group of 3 nitrogenous bases. And I hope you people know variable within the RNA have U, C, A, G. Okay, these are the bases, nitrogenous bases, those are available in the RNA. I explained you in the previous lecture, guys. Thymine is in DNA, but thymine is absent in the case of RNA. In place of thymine have a presence of uracil, the UCAG in RNA. Okay, so total how many genetic codes are in the RNA? Inside the human body. So as you remember, not only human body, every organism have a 64 genetic codes in the form of triplets. Triplet means group of 3 bases. The picture will be very clear in the next PPT. Wait, guys. And again I repeat, guys, total amino acid in the human body are reported are 20. So whatever the protein formation happen, this all the protein formation of the human body is synthesized through the help of 20 amino acids. So total number of genetic code, 64 genetic code. Total amino acids are 20 amino acids. So 64 genetic code are representing the 20 amino acid, and these 20 amino acid in many of the amino acid in different sequences, they form the different type of protein. This picture I'll explain with you. Just wait, guys. But the total genetic codes, those are available. Okay, they are 64. Man who got the Nobel Prize behind the discovery of genetic code, you can see on the screen, Nirenberg, Mithai and Khurana. So Nirenberg, Mithai and Khurana, they got the Nobel Prize for genetic code. You should know, guys. What about the feature of genetic code are codons? See, genetic codes are codons, the features of it. Okay. Actually, you know what happened? Genetic codes are codons are in triplets. Triplet means a group of 3 bases. Okay, one thing more everybody should know, genetic codes are in RNA. Again, I repeat, genetic codes are in RNA, and RNA have these bases U, C, A, G. These are the nitrogenous bases. And they're in the triplet, the group of 3, either this, yeah, any other arrangement. But there will be always in the 3 in the group, guys. Okay, and genetic codes are of 64 types. Okay, you just come here, guys. Genetic codes, this is the genetic dictionary in the screen, guys. Later on I will go in back. Actually, this is the genetic dictionary. Okay, that is behind the genetic dictionary, Nirenberg, Mithai and Khurana. These all the person name you have seen, those got the Nobel Prize. In the genetic dictionary, what you will do, guys? See, there is a 16 boxes here. See, there are 16 boxes here, and within each and every boxes have a what? 4 boxes. Just like, see, one box. You see what I am doing, guys? Okay, within each and every boxes have another box. See, another box. See, another box. So what you got it? Within the each and every have a 4 boxes. In that way, 4, 16 have a 64. I hope you understood. Similarly, the boxes will also be here. Okay, in that way, guys, total how many boxes you will get in the genetic dictionary? 64. And each and every boxes have a genetic code are codons. Those are in the triplet. Triplet means group of 3 bases. As you, in every boxes you can see have a 3 bases are the, see, 3 bases. What a genetic, Nirenberg, Mithai and Khurana explained. They said, like in this genetic dictionary, how the genetic dictionary will complete. The first of the letter you write this in the box. Okay, second side the letter you write this one in the box, and third side is the letter this one you write in the box. So what will be the first genetic code in the genetic dictionary? See, the first letter is this, U is here. Second letter is upside. See, the second U is here, and the right side is the U is here, the third is here, and this U is the first genetic code in the genetic dictionary. Everybody clear? See, say another side. First letter is this side, left side. So this is in the boxes is U. The second upside is U, and the right side is what? This is a C, guys. So that is the C. UUC is the second genetic code. I hope you understood in the genetic dictionary how this genetic dictionary will complete. If I ask you guys in these boxes, what should be the genetic code? See, the left side have here is what? C. In this box have a C. Upside you see there is C. So this is C. CC. And the right side is what? That is U. So this is one more genetic code in the genetic dictionary. If any of them I ask you, these all 64 types you will get here, guys. And you know what is mentioned here in this case? Whatever the genetic code form UUU, UUC, UUA, UUG, CUU, CUC, and see these genetic codes are representing these various type of amino acids. Those you can see. Total number of genetic code is 64. If you count the amino acid 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 11. Stop codon not synthesize the protein. 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Why I'm telling you more than 20 is looking like some of them are repeated. Okay, here you can find and the same name you will get it. So in that way, total 20 amino acid are repeated. See, stop codon. Stop codon are not responsible for the synthesis of any proteins. Okay, stop codons are not responsible for the formation of any protein. So this 64 genetic code, as I explained you, 64 genetic code represent 20 amino acids. How should 20 amino acid? The very simple, guys, maybe one amino acid, just like phenylalanine you see. Phenylalanine, maybe one amino acid have a one or more than one genetic code. As in the case of phenylalanine, you people can see. As in the case of phenyl, see guys, just again come back. Just in the case of phenylalanine you see. In the case of phenylalanine, you will see this amino acids have a 2 genetic code, and I explain you 64 genetic code is equal to 20 amino acids. 20 amino acids. I hope everybody clear, guys. Okay, everybody, see here, this is the this one amino acid have a 2 genetic code. See, this is the leucine. Leucine have a 6 genetic code, 6 genetic codes. Either UUA, UUG, CUC. So any of them in RNA, when they synthesize the amino acid, it is a leucine. So this is the phenylalanine 1. This is the leucine, the second amino acid. Isoleucine, methionine, valine, okay, serines, means these are the actually 20 different type of amino acid. Tryptophan. Stop codon, you remember, stops are those codon that I'll explain with you, which never synthesize any kind of protein. That's why here have no amino acid, no amino acid. Okay, arginine is here, arginine is here. So that's why the 64 genetic code have a 20 amino acids. I hope you people very clear. And now I'm coming into the feature of genetic dictionary. You will be very clear here what are written. So the codes are in triplets. Now the picture is very clear. This was the question in MPPSC 2000, MPPSC 2020-21 as examiner asked. Okay, it is in triplet, a group of 3 bases, you know very well. When the protein synthesis start, so there are some places from where the protein synthesis start, that is called initiation code. Okay, so inside the RNA, suppose this is the RNA sequences, these are RNA sequences. If suppose this from here the protein synthesis is going to start, so definitely here the sequence should be AUG or in some cases GUG, and then from here the protein synthesis will start. So AUG is responsible for the formation of amino acid, that is methionine. So whenever any work is going to start, so the work is going to start with the AUG. AUG for the methionine amino acid is written here, guys. Okay, everyone, now come. Next one, guys, whenever the protein is going to stop, the protein is going to stop at some codon UAA, UAG and UGA. This is called as a ochre, UAG is called umber and UGA is called as a opal. And after it there is no synthesis of protein, and now the chain stop for the protein synthesis. Okay, everyone, and these stop codons you people have seen here in the C stop codon UGA, see UAA, UAG, UAG, and you see what is here, UAA, UAG and UG. I hope you people very clear. What is the meaning of degeneracy of codon? Degeneracy of codon means a single amino acid coded more than one codon. You already have seen. A single amino acid coded. See, a single amino acid coded more than one codon. See, leucine coded 1 to 6 type. I hope you people very clear. This is the reason I told you first you see genetic dictionary and then I will tell you the genetic dictionary property. Okay, what is the universality of codon? Universality of codon means the same genetic dictionary and codon, this genetic code represent for this amino acid, it is in every organisms. You, plants, everyone have a same symptom. Dog, cat, lion, monkey, chimpanzee, everybody have a same symptom. Comma-less codon, no comma use in these between 2 codons, you already have seen. No overlapping codon. What is the meaning of no overlapping codon? Suppose this is RNA, guys. 1, 2, 3, 4, 5, 6, 7, 8. Suppose some bases. A, U, C, C, A, C, G, U is there. Suppose this is one of the genetic code. Okay, this will be the second genetic code. It is not like that this will be the overlapping of genetic code. This is the basis that will overlap from this and this. No. Exactly, this is no overlapping codon. Neither if here in this 3 bases the G is there, in these 3 bases G is there. So G will be overlapping. No overlapping codons there. This 3 is independent and this 3 is independent. The code is a non-ambiguous. The code is a non-ambiguous. What is the meaning of the code is non-ambiguous? Thus one codon cannot specify more than one amino acid. The one codon cannot specify more than one amino acid, you must be clear. One codon means genetic code cannot specify more than, see, one amino acid can specify more than one codon, but one codon represents only one amino acid. See here, guys. If I ask you UUU represent which amino acid? Only phenylalanine. Phenylalanine can represent many genetic code, but UUU only phenylalanine. UUU not responsible for leucine or anyone. So that is the reason I am telling you here the codon is non-ambiguous. The codon is non-ambiguous means one codon cannot specify more than one amino acid. Only one codon represent one amino acid. So I hope you people very clear with the genetic dictionary concepts. Now I'll come into the cell cycle. This is also very important, guys. Whenever the cell is going to divide, there are few phases the cell is going to pass, and by cell cycle and cell division is important. Very simple, guys. Because of cell division, an organism will grow. So growth of any organism is because of cell cycle. So the growth of any organism is because of cell cycle. Okay, and within the cell cycle have 2 phases, guys. The first phase is the interphase and second phase is the M phase. Whenever the cell division perform in the cell cycle, cell cycle means what? Cell divide, again cell grow and further cell divide. It's a kind of cycle. So in the case of cell division, when the cell cycle happens, here cell division happens. Within the cell division have overall, if you see in the cell cycle, have two phases. The one is the cell cycle have a two phases. The one is the interphase, second is the M phase. Interphase is a kind of a pre-division phase. Pre-division phase you can call as a regulatory phase and control phase. And M phase is called as a division phase. M phase is called as a division phase or response phase. So I hope people very clear. Whenever the cell is going to divide, so cell is going to pass from two phases. The one phase is called interphase and second is called M phase. Interphase is a regulatory phase and pre-division phase, this is also called as a control phase. But M phase is finally division phase. Okay, interphase is more important, guys. The reason how a cell will divide in the M phase, every regulation and controls are under the hand of interphase. Disturbances of interphase can cause a cancer. The reason the division phase is totally disturbed. And the division phase is going to disturb, so it is out of control, and this will be the problem here in this case. There are two types of division, as you can see here, guys. In the M phase, the division phase of two types. Here you can see two types. The one is called as a mitotic division and second is called as a meiosis division. So mitotic or mitosis, you can also call it as a mitotic or mitosis, both are same. Mitosis division was given by Flemming. In this case, somatic cells are vegetative cells. They have a mitotic divisions. Okay, you know, somatic cells and reproductive cells, I told you in the previous lecture, guys. Reproductive cell is your testes and ovaries, and somatic cell, a part of, see again I'm telling you, there are two nature of cells. The one nature of cell is called as a somatic cell, and second nature of cell is called as a reproductive cell. And second nature of cell is called as a reproductive cell. A reproductive cell is also called as a germ cell or generative cell. This is a germ cell or generative cell. Actually, the somatic cell involved in mitosis division, you must be know. And reproductive or germ cell or generative cell responsible in the meiosis division. In the case of mitosis division, what happens, guys? Same number of chromosomes produced. In the previous case, I told you, we people are the diploid, guys, in the previous lecture. Okay, again I repeat my words, guys, don't jump. Kindly listen my all the videos in the series, then the coming series will be beneficial to you. The reason the whole entire discussion I am doing in the cascading manner, not random as you people are preparing. And this is the main problem in UPSC preparations, those people are doing. Okay, kindly follow from the beginning, guys. Okay, if you are listening the first time directly the lesson number 5, so it's not fine. Better you start from lesson number 1. So we people are the human beings are diploid. Diploid means 23 pair of chromosomes. When the cell divide and form 2n and 2n type, this type of division is called as a mitosis division. But if a parent cell is 2n type, and after division they form the half number of chromosome n, and this type of division is called as a meiosis division. So what happened in the case of mitosis division? If a parent cell have a 23 pair of chromosome, after division 2 cell or 4 cell form, they again have a same number of chromosome, 23 pair and 23 pair. But what happened in the case of meiosis division? If a parent cell is a 23 pair, after division they form 23 and 23. This is called meiosis division. So what actually mitosis and meiosis division? You people very clear. Okay, brilliant. The mitosis division occur in the somatic cell. The picture is very clear. Somatic cell are vegetative cells. Here the two daughter cells, those are formed, they are identical to their parents. Definitely, guys, because in this case have a no crossing over. The exchange of genetic material will not happen. That is the reason whatever the parent cell have, the same type of cells you will get in the case of those cell produced here. Okay, you remember, guys. But in the case of reproductive cell, those cells produced here, they are not like to the parent cell. The reason here the crossing over happen, there is crossing over has some process that is called exchange of genetic material. So overall you remember somatic cell, this is the copy of parental cell. But reproductive cell is not a copy of parental cell, you remember. And here in this case the chromosome number remains same in the daughter cell. So this is the parent cell and these are the daughter cell. In the mitosis division, what happened, guys? Those happen in the somatic cell, number of chromosomes are same. So now I hope through the help of this picture everybody very clear. Okay, so where is the reproductive cell? I hope you know very cell size. Reproductive cells are available inside your testes or inside your ovary. It is a reproductive part. And many other name used for the reproductive cell. This is called germ cell, GERM, or generative cell, GENERATIV. And here the germ cell or generative cell have a meiosis. What is the role of germ cell or generative cell? Definitely, guys, at the time of reproduction. Okay, at the time of reproduction, they produce a half number of chromosomes. This is the reason I told you at the time of reproduction from the father side, when the testes is going to generate the sperm and ovaries are going to generate the egg, they have a half number of chromosome. Testes is 23 chromosome, 23 pair chromosome, ovaries 23 pair chromosome. But when they release the sperm, guys, in this case, meiosis division happen, and because of meiosis division, number of chromosome become half. The same thing from the ovary side. Mother ovaries are diploid, but when the mother ovary involved in the formation of egg, here the meiosis division happen and the number of chromosome in egg also become half. Very interesting topic, guys. Try to understand. Now this is meiosis, and see this whole entire discussion those I'm doing with you guys, this is very important discussion that will connect with you in the case of biotechnology. So this is the reason those all the videos you people are listening me here on the YouTube, this you people always save and always listen, guys. It will build up your concept to understand my biotechnology classes, those I'm taking on the advanced level. In the plus class courses, those I'm taking the biotechnology, without the help of these all the lessons, you people will not be clear. Okay, so now come the second type of division, meiosis or meiosis division. So it's my humble request to all of you, those people are listening my plus courses, especially my plus courses for science and tech and environment. I'm telling you, whatever the videos I'm making on the YouTube, kindly listen it, guys. I know your time has a value, guys, and I also never like to waste your time because my time have also my values. Okay, still I'm busy a lot of in the face-to-face classes. I'm taking your classes. The reason I know very well, guys, every platform have their own importance. Either it's a face-to-face or online. Because many of the students, those have not ability to reach in the institutions, still they are listening all the videos online. So here I like request to all of you guys, don't miss to listen my advanced courses those I'm taking on the Unacademy right now. Okay, right now I'm associated with the Unacademy. Here all the advanced classes of science and tech and environment also I'll tell you guys. The reason these are those subjects where you can find out 30 plus questions very easily, and 20 easily you people can find out. Okay, maximum people, guys, you know very well, guys, my 2 books are Science and Tech Environment on Unacademy, on McGraw-Hill, that I request to all of you. The Science and Tech came 5-6 years back. Today is the 6th edition, is coming in the market. Okay, and the science and the environment have a 2nd editions, and every time it is updated. The reason I'm connecting with this classroom program like Unacademy face-to-face classes and you know the updation of the books, so that is the reason my course and my book you just follow, and my all the YouTube sessions those I'm taking on Unacademy, so your integrated preparation will happen. Okay, so whatever you feel, guys, accordingly you follow, but I request to all of you, kindly listen the science and tech environment at least once, whatever I'm taking the classes, and listen the books once again. I request, you will definitely get the reason why I'm telling you. Okay, the term meiosis was used by farmer Murray. Reproductives, you know very well, that is in the reproductive germ cell. Now everybody know our haploid cells are produced, the 23 chromosome, 23s are diploid. Okay, this term is used for the diploid in the previous lecture, and when they form N and N, ultimately it is the haploid. So here the haploid cells are produced. The chromosome number reduced, half 23 chromosome, pair chromosome become here 23 and 23. The significance of meiosis, I told you here, the recombination happen. Recombination means crossing over happen. Crossing over means what? Crossing over means exchange of genetic material. Exchange of genetic material means those genes, exchange of genetic material, those genes are available in the parents. Okay, you people are getting the crossing of the gene, guys, in the next generation, and that is the reason you people are not look like 100% your papa and mama. Your characters have somehow some papa and some mama characters. Okay, and that is the reason here I mentioned with you, due to crossing over, and the number of chromosome become half, everybody know, from the parent cells. One of the interesting topic, that is a miscellaneous type, I think so in the very advanced level of questions, if anywhere it is coming, like CDS, NDA, UPSC type of questions, the colchicine may come. Actually, you know, colchicine is a chemical. You should be know. It is a kind of alkaloids. It's a chemical, but alkaloids. This alkaloids chemicals is obtaining from a plant that is called Colchicum autumnale. Colchicum autumnale belongs from a family Liliaceae. Okay, it induces the polyploidy. Polyploidy means suppose a plant is a diploid nature. If we use the colchicine powder, the colchicine powder will multiply this 2n and become as a 4n, and because of this the characteristics of the plant will become gigantic. I'm asking you guys, in the case of me, through colchicine this polyploidy is not possible. Polyploidy means what? One side is a haploid, second side become diploid, it becomes a tetraploid, octoploid, ultimately is called poly. Poly is multiploidy you are going to increase, and see whatever the characteristics in the n, in the case of 2n the character is double. In the case of 4n, the character will double of 2n and the 4 times of n. So ultimately, sir, you are saying the organism size will more and more. Yeah, because number of chromosomes are more. The characteristics of organism are more. Yes. So this is called as an induced poly, but in the case of plant it is allowed. Not every plant, but most of the plant you can do it. In this cases, what happened, guys? In this cases, the plant induces the polyploidy, and plant will have a more and more better characters, more and more productivities in this cases, guys. But it is not universal truth that in the every plant, whenever we are doing the polyploidy, the characteristics will become double. But happened. So in the case of plant breeding, guys, when we are trying to increase the plant characteristics, the plant productivities and all, so the scientists are doing this colchicine activities. Actually, the colchicine stop the formation of spindle fiber. Spindle fiber is one of the centrioles type of cell organelles. Those are responsible to separate the chromosome within the cells. And that is the reason, guys, chromosomes number become double. Okay, whenever the 2n cell is going to form 2n and 2n in the case of meiosis. In the case of mitosis. So and inside the 2n become even double and become 4n. Inside it happened, and then it half and half separate, and again 2n become 2n and 2n. I hope you can understand. But in the case of polyploidy, this 2n when become as a 4n, when it is going to become as a 2n and 2n, in that case when the 2n diploid, it not form the cell plate and not two cells separate as 2n and 2n, automatically it will become as a 4n, because the spindle fiber are those separate the chromosome 4n half and half, and then two cells separate. This is the reason here have a diploid formation, tetraploid formations. Okay, see, X-ray and UV rays inhibit or retard the growth of cell division. If somewhere the scientists want to do to reduce the growth of any particular sites of the plants and all, the X-ray and UV rays you can use to retard the growth. As the colchicine or multiply the growth of organisms, here the cell divisions can stop. In this case, some of the antibiotics you can use like chloromycetin, tetracycline, butyramycin. They block the cell division, interfere with the protein synthesis. So this was the entire discussion. I hope you will enjoy a lot of the knowledge you got it in my lessons here. So it's my Telegram channel where I'll tell you to all of you where you can find out all the entire PDF. And Unacademy profile is unacademy.com, Ravi P Agrahari 2010. Here you can find out my all the plus classes, those I told you here, guys, where you will find out my all the advanced level of discussions. Ravi UPSC is my code. Don't miss it to use it whenever you are going to join any courses. Ravi UPSC code will give you extra 10% discount. UPSC CSE test series for the mains examinations are running now. That is for the general studies and optional both. Whenever you are going to join, kindly use the Ravi UPSC code. CSE Store is the option that Unacademy is giving you if you are going to buy a courses of 2 years and above subscription. The extra free 1 year extension or 2 year extension Unacademy will give you in the case of your requirement. Ravi UPSC is the code. Kindly use to join this course. Why Unacademy is the best option? Widest choice of educators, flexibility options, unlimited views of lectures. Live mentorships, 178 plus toppers got select on Unacademy with the help of Unacademy in 2020-21. 0% EMI facilities on Unacademy also available. Here you can find out loans. UPSC CSE next batch is going to start from 21st July. Don't miss to join it, guys. Here for 2023 preparation you can do with all the experienced faculties on Unacademy. Bilingual and Hindi batch is going to start from 21st July. The courses of Science and Tech Environment are running now on the plus courses. Kindly enroll now with the Ravi UPSC code. And thank you everyone. Kindly subscribe the channel, like and share this video. Download this PDF and connect with me always. I'm always with you guys. Regarding the updations and connection with me, you people can send me a message on my telegrams as this number is available everywhere. Thank you everyone. God bless you. Meet you soon in the next class. Take care of yourself. Jai Hind.